multiple workflows we have here that allow us to comprehensively characterize a tumor. And each of these is optimal for a different type of question and a different type of therapeutic modality. I'll start with discovery-seq, in which we can take an FFP sample or a frozen tumor sample and comprehensively assay over 10,000 proteins in a very small amount of biological input. Next, we have our target-seq, which allows us to take that same biological sample and in a very precise and sensitive and quantitative manner, assay one to a small handful of proteins that are of highest value for you, whether it be a target engagement marker, a pharmacodynamic marker, or any other related protein. On the other side of this, for particular therapeutics, including those that target cell surface proteins, such as ADCs, T-cell engagers, radioligand pharmaceuticals, CAR therapies, or vaccines, we have our surface-seq workflow, which allows us to tag the actual surface proteins in a human FFP or frozen tumor sample, extract those cell surface proteins, and differentially measure their abundance both in tumors as well as normal tissues as a means for identifying new targets and validating those targets or stratifying patients for particular therapies in that modality. We have what's called our immune-seq and resistance-seq workflows, which allow us to understand specifically those proteins that are related to immune biology or resistance biology in tumors. And finally, we have what's called our signal-seq workflow, which is a workflow that's optimized for phosphoproteomics, which allows us to take those same FFP or frozen tumor samples and comprehensively identify the phosphopeptides and kinases that have been activated or repressed as a means for understanding the pathways that are active in a tumor cell. Yeah, it's a really great question. And genomics has transformed our understanding of tumors, both DNA sequencing, in which we can begin to sort of understand and map the mutational landscape of tumors, as well as RNA sequencing, which allows us to understand which transcript have been turned on and off in particular tumor cells, either on a bulk level, a single cell level, or on a spatial level. Now, once again, for the majority of tumors, 60 to 70% of them, there's not a single activating oncogenic mutation that can be identified, or RNA sequencing fails to accurately quantitate those surface proteins that may be critical for, say, an ADC. This is where proteins are absolutely essential to be able to assay as part of a molecular characterization of a tumor. If you recall, most therapeutics today work by binding or interacting with an actual protein. Typically, we're using DNA or RNA measures to infer those protein levels. Now, for certain proteins, this works well, but for the vast majority of the proteins in the proteome, this is inaccurate at best. Now we finally have the means to accurately quantitate the proteins, whether it be on a discovery level for targeted proteins or tagging and measure surface proteins, for immune proteins, resistance proteins, or signaling proteins. And that really represents the gap here that we're closing by the application of these proteomic technologies. So the way these emerging therapeutics, particularly these precision-based therapeutics operate, whether it be antibody drug conjugates or ADCs, T-cell engagers, radioligand pharmaceuticals, or CAR therapies, are by engaging a protein that's present differentially at the tumor cell surface, but is not present in normal tissues. And in doing so, they're taking advantage of this differential expression at the cell surface to selectively target a cancer cell while sparing a normal cell. Now, in order to do so, we need to be able to identify what proteins are at the cell surface and to quantitate them in a manner that allows us to understand which ones are present in tumors but not normal tissues. This is fundamentally what surface-seq does. We take advantage of unique post-translational modifications that are present on tumor proteins that are present at the cell surface, and in doing so, tag those proteins and then pull them out and assay their abundance by mass spectrometry. Typically, for a tumor, we can quantitate somewhere in the order of 1,000 to 2,000 of these cell surface proteins and measure those same proteins in normal tissues. When we do this, we can identify new targets for ADCs or T-cell engagers, new receptors for radioligand pharmaceuticals, or new antigens for CAR therapies and other related cancer modulators.